Hello, everyone, and welcome to today's webinar, Single-Cell Multiomic Characterization of Pediatric Gliomas, Uncover Novel Subtype-Specific Therapeutic Approaches. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Scale Biosciences. To learn more, please visit scale.bio. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would now like to welcome our speaker, Dr. David Rincón Fernández Pacheco, Project Scientist, Board of Governors, Regenerative Medicine Institute, Cedars-Sinai Medical Center. For a complete biography on Dr. Rincón, please visit the Biography tab at the top of your screen. Now, before we dive into Dr. Rincón's presentation, we also have joining us for a brief introduction to Scale Biosciences Technology, Archana Beditapur, Product Manager of Scale Biosciences. Archana, welcome. You may now begin your presentation.
Hi all, my name is Archana and I'm excited to talk to you about single-cell biology at scale. Single-cell genomics has been a critical technique in drastically increasing our understanding of human health and disease and all of its complexity. Methods have been developed to assay across the spectrum of biology from the genome, epigenome, transcriptome, and proteome, but measuring these modalities in small subsets or small sample numbers places a ceiling on the next level of insight for greater impact. Scale's mission is to get to that next level of scale for cells and samples for deeper insight into biology. The single-cell field is only growing as researchers have realized the need for single-cell granularity complemented by high sample throughput and easier sample preparation in a number of research areas to enable longitudinal studies, patient cohort diversity, rare cell population characterization, and training for machine learning models. Scale's massively parallelized barcoding technology allows researchers to get high-quality single-cell data with flexibility in workflow and ability to scale up cells and samples, which is actively being expanded to new modalities. Single-cell methylation is a great example of that product innovation. As a recent entrant into the single-cell sequencing space, we are keeping a high pace of innovation as a company. Within the past 2 years, we have launched 4 products to enable researchers and plan to further expand our portfolio with impactful and differentiated products. In this webinar, we will be discussing single-cell methylation on the right here. We put a significant amount of development work into making our workflows as streamlined and user-friendly as possible from start to finish. The fixation that enables our massively parallelized barcoding also allows for experimental flexibility, where samples can be stored and shipped on dry ice after locking in your biology. Our workflow is plate-based, and reagents are pre-stamped to minimize pipetting. This eliminates the need for specialized instrumentation and enables broader compatibility with automation. We have incorporated smart plastics for ease of use with a pooling funnel, which removes the need to do pooling steps with a multi-channel pipette. You simply flip over the 96 or 384 well plate into the funnel and give it a gentle spin down in the centrifuge. Prepared libraries are compatible with Illumina sequencers, and all of our workflows have fully supported bioinformatics pipelines to process your sequencing data, leveraging cloud-based platforms and compatibility with advanced downstream analysis tools. Now, I'd like to give an overview of our single-cell methylation kit and workflow. We are excited to enable single-cell DNA methylation, as it is a well-studied epigenetic mark typically done in bulk or arrays today, and this is the first commercially available methylation kit on the market. Our technology enables single-cell methylation by combining bisulfite conversion with Scale Bio's massively parallelized barcoding, which uses the cell as the compartment. Fixation and permeabilization-enabled cells or nuclei are used as the reaction vessel. Sequential analysis of short DNA tags to the analyte of interest creates a unique index per cell. Combinations of indices and number of cells or nuclei scale exponentially, removing the limitations of Poisson distribution. This allows us to vastly increase the throughput of single-cell methylation compared to homebrew academic methods. In addition to the kits themselves, we also provide the option to run your samples through our in-house services. This can be for proof of principle or even whole projects, depending on the resources available in your lab. Our website is a great place to go to get more resources, more information on our products, technical notes, webinars, and access to product support.
Thank you, Archna, for that introduction into Scale Biosciences technology. And David, welcome back onto camera. Thank you again for joining us, and let's get you started on your presentation. You may now begin.
Hello, everyone. Today I'm going to be presenting some of the results that we are generating in our lab using different multiomics technologies to try to tackle the question of how tumor distinguish and how the heterogeneity of tumor is derived. So to give a little bit of background in brain tumors, this type of cancer can be classified in different ways depending on what you want to look for. You can look for DNA methylation, and you can see in this second row in the plot, which has been recently one of the most relevant ways to classify tumors. And you can try to figure it out different mutation and see the driver mutation of this tumor that you can see by columns. What I want to show in this plot is how heterogeneous brain tumors can be, which is really important in terms of treatment design and how you tackle this problem. Now, if it's not only inter-tumor heterogeneity, which is a problem, because even in the same tumor have been proven that you can find different clones that are going to have different driver mutations. And that means that they're going to respond differently to the treatment that you are giving. And in our case, we are working in pediatric brain tumors. And for historically, they have been just classified and treated in the same way that adult brain tumor. But we now know that they are completely different, to the point that the World Health Organization make a new classification just for them to have the pediatric brain tumors separated from adult brain tumors. And they can be classified depending on their, if they have mutation in the histone tree, which is the most common mutation you can find in these tumors, or if they don't have those type of mutation and they carry other mutations. So that's how they did this classification. I hope all of this big heterogeneity can give you an image of why we need better models. And that's what we thought in the past when we think like, okay, we need to figure out a way to analyze these tumors. So to do this, our laboratory built the mother reaction, the mother technology, which is a mosaic analysis by dual recombinant mediated cassette exchange. And the way it works is using a Cree reporter mouse, the ROSA 26 MTNG mouse, which the usual way it works is just you have this locus here in the ROSA 26 locus, you have the cassette of the TD tomato, which is expressing TD tomato constitutively. And when you backcross this mouse with a mouse expressing Cree recombinase in a specific tissue, those cells that are having the Cree recombinase expression will flux out the TD tomato, and those cells will become from red to green. So this is the usual way that the system works. But the good thing is that these cassettes not only have the logs beside, which are the green triangle, the yellow triangle here, but also have sites for another recombinase, the flipase recombinase, which is the FRT site, which are in blue in this plot. And the good thing is that you can combine those two recombinases, and what they will do is really interesting, because they will be able not only to remove what is in the cassette, but also make a recombination with a plasmid donor that you can introduce. So what we do with the mother technology is that we put another plasmid that it will have flanked by the LOXP site, and the FRT site will have a gene of interest that we want to introduce, in this case, the oncogenic driver of our tumor models. And when we add the recombinases, these recombinases will just switch the cassette, the internal cassette, the locus cassette, with the plasmid cassette. That this way you will have your cells expressing your oncogenes in just one location of the genome, and you won't have, like in other systems, thousands of copies like with your transposon or viruses to introduce your oncogenes. So using this system, we have been working on it for a long time now, and we have been able to generate what we call the growing zoo of mother brain tumors. So as you can see here in this summary, we have generated different type of histone 3 mutation, a pediatric brain tumor. We have generated ependymomas related with fusion protein, like the CFTA relay or the JAK1 MAM LD1 that you can see in the figures on the right. And even we have generated adult brain tumor with the EGFR-B3 mutation. Now, for today's talk, we're going to be focusing on these three tumors here, which are pediatric brain tumor, and the only difference between them is just one amino acid. So you will have the histone 3 with a K27M mutation, or the same histone 3, which is going to have the G34 mutation, or the histone 3 wild type, keeping the other mutagenic gene, the PDGFRA and the P53 active. So for our project, for our model, what we did is to generate this P donor plasmid for mother reaction. As I say, we have the histone 3 marked with a fluorescent protein, a constitutively active PDGFRA, which is also a really common mutation in these pediatric brain tumors, and the dominant negative P53. And P53 is like the most common mutation you can find in any cancer. And what we do is use a neonatal electroporation to introduce this mutation in newborn pups and let them grow. And as we published in Cell some years ago, 5 years ago, with this system, you can generate the tumors that are actually replicating quite well the human tumor. So we have K27M tumor that grow in the midbrain and the striatum. We have G34R tumor that invade the corpus callosum and then invade the cortex, which is how this tumor grow. And then we have the wild type tumor, which show a mixture of phenotypes. And also you can see in the survival curve how the K27M are the more aggressive one, as it has been seen in human data. Now with this tumor, what we can do is to actually microdissect them and analyze them with single cell modalities, with different single cell modalities. And that's what we did using all this plethora of different packages that now we have in R and Python. So our first approach was to look at the single cell RNA-seq. And this is an image of what we see here. So you can see on the left that we have a tumoral mass which have a cycling population in yellow on the bottom, an oligo-like cells, which are a really common program in gliomas and brain tumors. Then an astrolite, which is the small corner to the top right. And the vast majority of the mass of the tumor will be OPC-like, oligodendrocyte progenitor cells-like. And then we have all the other surrounding cells in the microenvironment that you can see, the immune system, microglia, peripheral macrophages, T cells, B cells, and then the endothelial cells, pericytes, and other type of cells that are going to be there, like, for example, the coloplexum. The interesting thing with our methodology is that since we are actually introducing a different gene, which is going to generate a different transcript, you can look for that in your single cell RNA-seq and see how it looks like. And you can see that our transcript, our oncogene is expressed basically only in the tumoral cells, while the endogenous normal transcript, which is going to be the 3D tomato expression, is expressed in any other thing. So it's a really nice way to sort out tumor cells from non-tumoral cells. But the most interesting thing for me, and what was really striking, is that these three tumors are completely different in their phenotype, what they do, how they grow, spatially, temporally. But at single cell level, at short-read single cell, I must say, they look completely the same. They completely overlap. So to try to look into this and see if we can find these differences, the first thing we did is like, okay, let's remove anything that is not tumor. Let's focus on the tumor and see how they look like. And you can see that they completely overlap. So to try to find different transcriptional program, we use RNA velocity, which is an algorithm that takes care of splice and non-splice sequences to try to figure out the transcriptional pattern that these cells are following, the evolution of these cells. nowadays it's a little bit controversial, but in our case, what we were more interested in it was the most stable transcriptional profiles, which will be at the end and root of the pseudotime that this algorithm creates. And here we can see in yellow all those stable transcriptional profiles. And when we look at them, we see this this transcript that they are expressing. We can try to figure out what they are. And we found the typical oligo-like, astro-like, the OPC, cycling cells. That's really common in the tumor. But then we found two novel transcriptional programs that I have seen explained before in other papers, but they have never been seen, or I have never seen them at single cell level, which is a myeloid-like population, which is expressing genes related with a myeloid pattern, and then a group of cells that are expressing a really high number of hits of protein on ribosomal protein. So I decided to look if these cluster, these cell types, were differentially distributed in our tumoral cells, and maybe that would be the difference between the tumors. And when I look at that, I've actually found some differences, but not massive, and something that are expected. For example, K27M cells have more cycling cells, which makes sense because those are more aggressive tumors, they are more proliferative tumors, so that makes sense. On the other side, G34R cells are showing a higher number of cells in the myeloid-like and the high hit shock ribosomal protein clusters, which are interested on its own, but is not so strong a signal, not so strong a difference to just figure out that that is the reason. So we went a little bit more close to the genome to see what is going on. So the next step was looking at single nuclei ataxi expression to see the genome. And when we did this, we didn't show anything really. We analyzed this data and we have some explanation here, but in general, single nuclei ataxi is giving us a less deep image of what we see in the RNA. You can see that the tumoral mass now becomes just a blob, which almost no different. We can figure it out poles of expression that we can see cycling cells, oligo-like, which is the most differentiated type of cell that we find in the tumor, and then a general tumor mass, but they are not really branching out. Then you can see the microglia, the immune cells, the endothelial cells, but not too much. And again, tumor cells from the three different tumor subtypes completely overlap between each other. So we decided to go a little bit deeper and look in the DNA methylation. And why we do this? Well, DNA methylation, it's really well regulated. It has been really well established that can be regulated by the histone 3, because the histone 3 is an epigenetic marker. And these mutations that we have are actually modifying marking on this histone 3, which is going to affect how they mark the DNA on its own. So we decided to look at this, and our first approach was to look at bulk DNA methylation. So we use an array of probes that cover the mouse genome. To do this, we use organoids, and we use organoids generated from our primary models of mouse brain tumor, because an organoid, when you culture them, you are going to wash out, not remove, but they don't divide so much, so the tumor cells are going to overgrow any other type of cell. So you are going to have, which is not an organoid anymore, but a tumoroid, because it doesn't have all the other cells. But it's really pure in tumoral cells, and we have checked at single cell level that they keep the internal heterogeneity that we see in the primary tumor. So we are not losing transcriptional profiles, but we are removing the non-tumoral cells since we are doing bulk. And in our first approach, we really clearly see differences here. So in the PCA plot that you can see in the bottom, you can see that the three tumor subtypes really separate between each other. They are not overlapping anymore. And when you look in the heat map of the methylated probes, you can see that the K27N is massively hypomethylated, which is expected and is already described. And the wild type and G34R tumors also show differential methylation region. So we decided to look in this hypomethylated signature, try to figure out which genes are actually getting altered by these mutations, these epigenetic changes. And as you can see on the bottom here, this is a dot plot taking those hypomethylation signatures, which are taking all those genes and making a pseudogene, a metagene that we can look in the single cell RNA-seq. And now, even if the single cell RNA-seq is not able to separate our tumor, these signatures are extremely strong and specific for our tumor. So the G34 methylation signature is specific and clear for only the G34, and the same with K27 and wild type. And interestingly, these methylation signatures are not homogeneous. It's not like, okay, all the G34R cells are expressing this hypomethylation signature with the same intensity everywhere, and it's not on the other cells. As you can see on the UMAPs to the right, these hypomethylation signatures are more or less in all the tumors, but they are in different intensity and in different regions, and that is interesting. Okay, looking a little bit deep in this hypomethylation signature, we can do pathway analysis. And with pathway analysis, what you can see is that the G34 hypomethylation signature is really enriched in the arginine and proline metabolism pathway. And the K27N is enriched in the hypo signaling pathway, and the wild type is enriched in the cardiac muscle contraction, which doesn't say too much in tumor, but it's also enriched in the JAK-STAT signaling pathway. And both hypo signaling pathway and the JAK-STAT signaling pathway are really entangled with cancer, because those are pathways that regulate proliferation, regulate apoptosis, regulate organ growth, differentiation. So all of those things are really, really important and usually altered in tumors. So it makes sense that those are there. But I was really interested in what is going on with the arginine and proline metabolism in the G34R tumor, because that's something that it wasn't expected. And what we found when I went back to the single cell RNA-seq and look at the expression of the genes that are responsible, that are inside of this pathway, you can see that aside of some exceptions, the vast majority of the pathway is inhibited. It's downregulated. And this was interesting. So I wanted to check in human data if we can find this too. So I went to the bibliography, the literature, and found some nice data set from Mario Suba and from Nada Javado. And I merged those data set with our mouse single cell RNA-seq to see how they look like, and you can see that they really can cluster together, so I can compare group by group. And again, what I found is that in human data, this arginine pathway is even more inhibited than what we saw in mouse. So this is really interesting, because we could try to use this even more if we look at the hypomethylation signature, the hypomethylation signature that we use. I'm going to go a little bit further here so I can explain it from here. You can see that these hypomethylation signatures are also, which were found in DNA methylation, bulk DNA methylation in mouse, and checked in single cell RNA-seq methylation of mouse, are also extremely specific in human data. So these signatures are there and are in human. And these heat maps on the right are showing how the human data and the mouse data match together quite well. So this heat map is showing different expression level, different clustering of tumoral cells, comparing K27N against glioblastoma, against IDH, astrocytic IDH, oligodendrocytic. And you can see when we put all of them together and we put our data set together with them, that they are really matching together. So our models are showing really clearly that they match, they mimic the human. They are modeling right this human pathology. So the next step was to look what happened with the arginine. Can we work with it? And to do this, since it is hyperregulated, these two genes here, we can see the ASL and AAS1 are downregulated. They are less expressed in the G34R tumor, which means that these two genes, which are responsible of making arginine in the cells, are not able to work anymore. So these tumors are dependent on the systemic arginine. They are not able to make arginine on their own, while the normal cells, arginine is an amino acid which is a non-essential amino acid, but usually cells don't make it on their own. They take it from the blood, that the liver is releasing arginine in the blood system, and they can take it from there. But cells can make arginine on their own, and that's not a problem for normal cells. But these tumoral cells don't have the enzymes to make arginine. So they are added to the blood arginine. So we can work with that, and we can try to remove the arginine. And in fact, there is already a drug from Polaris, the ADPEC20, which is a pergaminase, that degrade arginine into citrulline, so that it cannot work anymore. So we try this. We check how these tumors react to amino acid lack, to removing the amino acid, and you can see it in the lower plot how the G34R tumor have a dose response to the concentration of amino acid, while the K27M and wild-type tumor don't show that kind of response. In fact, the K27M show a higher cell death when they have more amino acid, which was kind of strange. Then when we move to use a specific arginine deaminase, so an enzyme which is coming from bacteria that is actually degrading the arginine in the media, the G34 showed a clear specific increase in cell death that doesn't appear in the K27N or wild-type cells. So these tumoral cells, these G34R cells, are sensitive to the absence of this amino acid, so we can use this as a treatment. Now, while we see this and we are coming from bulk DNA methylation, it is interesting to think, is this changes global? Are we going to see when we treat against the arginine, are we going to see the whole tumor responding to the treatment, or we're going to have differential pattern? And for this, we need to know if these enzymes are equally methylated, are equally epigenetically marked in the cells. And we cannot see this in RNA because the enzymes usually have a really low expression, and when you do a short read, a single cell RNA-seq, you have a really high drop-off. So we need to move to a single cell methylation to look for those genes specifically at single cell and see if this response is homogeneous or heterogeneous. So for that, we start working with a scale. And what we did is, okay, let's repeat the experiment we did with the bulk DNA methylation and see what we have here, because these organoids are going to keep heterogeneity. So we're going to see these cells, and we're going to see how they are acting over these genes. And this is the quality check data, how many cells we obtained. This is a first approach. We are planning on doing more experiments with this to have a better image. But till now, we have a nice number of cells that we can work with. So our first approach was, let's look at it. This is a reminding of how the single cell RNA-seq look like. Everything is overlapping, so you cannot see clear differences. But when we did a single cell methylation, you can see how clearly these three populations are separating. Now we have differential clustering, and they are not completely overlapping, so they are different at the DNA level, at single cell level. We can look that the coverage distribution is quite good, so we are confident with this data, and we can see how the methylation varies through clusters. So we have the cells responding different. They all have the same mutation because they are inserted in the genome, but they are having a differential response to this mutation. So you have different clusters answering differently. If we go to the G34R cells, which are the ones that we are most interested in, you can see this is only the G34R cells, single cell methylation clustering, and you can see you have different clusters. And when we look at the expression of the two enzymes that we are most interested in, AAS1 and ASL, you can see they are differentially methylated. They are not homogeneously, and you can see in the dot plot how we have some clusters that are actually methylating AAS1, while other clusters, specifically the cluster 6, is extremely high, methylating the ASL, but not as much as AAS1. Those two enzymes are necessary for making arginine. So you can expect that these two enzymes are going to these cells are going to respond differently to the treatment, and maybe this methylation pattern is going to change. So what we are working now on is we are making a new project where we are taking these tumors, we are doing it again, the neonatal electroporation to make this tumor at 90 days. We will start the standard treatment, the standard approach, and we will use a combinatorial method, and we will be using different chemotherapeutic, radiotherapy, combine them together, and then adding this ADIPEC20, this enzyme that degrades the arginine, to see if this combined treatment work and which one is the best approach. And we have already been funded to do this by the Cure Search Acceleration Initiative Award, and we are working on a clinical trial that we are going to be using this preclinical trial to clean up which treatments are the best possible for the children that will go in the clinical trial. And with this, what we are going to be doing is doing a multiomic characterization of this data, an immunohistochemical characterization to see how these tumors are responding to treatment, and normally the survival curve to see if it's actually improving the survival of these mice. And from this data, we are really interested in doing multiomics, and the multiomics that we want to do is the whole panel. We want to see a single cell RNA-seq, but we also want to see a single nuclear ATAC-seq, because those two are complementary and you can do multimodal in the 10x machine. But we are mostly interested, and we will be looking at the single cell methylation because it's the one that is showing us clearer differences, and we can look at the DNA better, and it's more specific and have less drop-off. And finally, I am interested in trying to integrate this data with other technologies such as single-cell cut and tag, and there is also single-cell proteomics that we are trying to optimize in the laboratory now. So all of this together should give us a good answer of what is going on with these cells and the treatment. Finally, I would like to thank you all for being here and listening to me. I would like to thank my laboratory to help me in all this work, and my mentor, Joseph Brunick, and to Scale Bioscience, to giving me the opportunity to show you my results. And with this, I will just open the panel for questions. Thank you very much.
Dr. Rincón, thank you so much for that excellent presentation. So I'd also like to, welcome Archana back. Thank you. Okay, so it is now time for our live Q&A portion of our webinar. Now, to our audience, if you haven't submitted any questions, please do so now. Simply type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So we have so many great questions already coming in. I'm going to dive right in here. Let's start with this one. How many cells, nuclei do I need per sample for the methylation workflow?
Yeah, I can take that one. So our methylation workflow is fixation enabled to allow for flexibility. And the recommended input to the fixation reaction is 650,000 nuclei per sample. However, the protocol can accommodate inputs as low as 250,000 nuclei per sample.
Very good. Thank you. Our next question. What about for the RNA workflow?
Oh, sure. Yeah, I can take that one as well. So for RNA, we recently released our ScalePlex multiplexing workflow, which allows for fixation inputs as low as 50,000 cells or nuclei per sample. In addition, as a side note, this protocol also enables multiplexing of hundreds or thousands of samples in a single workflow. And just to reiterate, this is for our single-cell RNA kit.
Excellent. Thank you, Archana. Dr. Rincón, did it surprise you that the methylation data showed clear clustering when the RNA data didn't?
At the beginning, yes, I must say it was surprising, but then you think about it and it kind of makes a lot of sense because, as I say, these mutations are actually targeting epigenetics, are targeting the DNA methylation, so are going to be really quite present there. And also when you are doing single-cell RNA-seq, and in our case, we were doing short-read single-cell RNA-seq, you have a really high drop-off in genes that are not highly expressed. While when you are doing methylation is measuring the DNA, so you're going to have 1 or 2 copies of the DNA, and it's not going to distinguish between high expressor gene and low expressor genes. So that way, the DNA is not masking the effect of this low expressor gene that can have a really high biological effect, such as enzymes, for example, in this case.
Thank you. Next question. What is the expected effect of the arginine starvation? Will the tumor adapt due to the lack of arginine?
Yeah, so that's what we are more or less expecting. So we are expecting that the tumor probably is going to have a first reaction where it slows down while it's adapting, but we are talking about cancer and we are talking about brain tumor, and those are quite strong beasts, if you want to say it like that. So we are expecting them to adapt, but that adaptation is going to come with a cost, and that's where the post-analysis becomes really, really relevant. If we can see how those cells are responding, we can see how they are opening other sensitivities, and we can then tackle those with new treatments in a follow-up.
Excellent. Thank you. Now, our next question, and Archana, this one may be for you. Is the methylation kit compatible with target enrichment?
Yes, actually, our methylation kit V1.1 is compatible with essentially any HiDCAP enrichment workflow. We have demonstrated target enrichment with the Twist Biosciences human methylome panel using the fast hybridization target enrichment workflow. This resulted in a reduction in sequencing, which supports profiling more cells, more samples for single cell methylation patterns, and more economical sequencing use.
Thank you. And I believe we have time for one more question. Are there data sets available to test the analysis pipeline?
Yeah, there are. We do have data sets available on our support website. So please do check out our website, email us at support@scale.bio, or reach out to your local Scale Bio sales and support team representative, and we can help you out.
Excellent. Thank you. Now, we do have a couple questions asking for contact information for the Scale Bio representatives. Archana, are you able to provide that information for our attendees?
Yeah, absolutely. You can reach out to us at support@scale.bio, and maybe we can, I think that information will also be included after the webinar. Also, if you go onto the support site, or onto our website, there are a few forms you can fill out to get in contact with us, and we'll be happy to put you in contact with your local representative.
Excellent. Thank you. And to our audience, I just dropped in a little broadcast message for you. So in the bottom right of your screen, you'll see that email support@scale.bio. So feel free to click on that and request information. Well, Archana, Dr. Rincón, thank you so much for joining us today. Do either of you have any final comments for our audience?
Thank you for listening.
Yeah. Thank you, Dr. Rincón, for joining us. And yeah, thank you, everyone.
Yes, thank you so much, both of you, for joining us today and for your important information. This was an excellent Q&A session. I do want to thank our audience for joining us and for their excellent questions. We had several questions that will need to be answered via email, so we do have your contact information. And additionally, you can submit questions during the on-demand period. So come back, type your questions in the ask a question box, click submit, and your questions will be answered via the email address you provided at the time of registration. So I do want to thank Dr. Rincón for joining us. Archana, thank you again as well. And I want to thank LabRoots and our sponsor, Scale Biosciences, for sponsoring today's webinar. Now, today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So to all our friends out there, thank you for joining us. We hope to see you again soon. Have a great day, everyone. Bye-bye.